This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Broad-sense heritability is a very useful concept, especially in clonally reproducing organisms, where the parent transmits its entire genome to the offspring. In sexually reproducing plants, however, because of the independent segregation and assortment of alleles during sexual reproduction, not all of the genetic variance is heritable. This is because alleles can interact with each other at a given locus or between different loci. A given phenotype is the result not only of the additive effects of the alleles, but also of the dominance between alleles at the same locus. If there are dominance effects, it is impossible to estimate the additive effects of recessive alleles unless they are in a recessive homozygote state, because they are masked by the expression of the dominant allele. Similarly, the interaction between different genes, or epistasis, can prevent the expression of some alleles. Here, the inhibitor produced by the yellow gene prevents the expression of the purple color gene, and thus we cannot deduce the actual genotype of the plant by simply looking at the color of its flowers. So unless a parent can transmit its whole genome to the offspring, as is the case in clonal reproduction, the dominance and interaction effects are not heritable. These effects are lost during meiosis, when haploid gametes are produced, and new dominance and epistatic effects are made when the gametes of the two parents are combined together to form the new diploid genotype of the offspring. Thus, the only thing that the offspring can inherit from each of the parents are what we call the additive effects of their alleles. If you have the broad sense heritability for a sexually reproducing population of plants, it might not be a good predictor of the efficiency of a breeding program, as broad sense heritability does not distinguish between additive and non-additive genetic effects. So broad sense heritability is actually the sum of additive, dominance, and interaction variance relative to the total phenotypic variance of the population. But only additive variance out of these components is heritable. This is where the concept of narrow sense heritability comes into play, which similarly to broad sense heritability is the amount of genetic variation relative to the total phenotypic variation, but only for the additive effects of the alleles. Knowing the additive variance is key for estimating narrow sense heritability and reliably predicting the outcome of a breeding program. But it is impossible to distinguish additive from non-additive variation by simple observation and measurement of the populations, even if we control for environmental effects.